Okay, good evening. My name is Alex Binstock and I'm the organizer of the seminars, ELSC seminars. I'm from Odessa, that's why you rarely can see me here. But I came here today to introduce today's speaker, and for me it's really great pleasure to introduce Mike Gutnick. So I know Mike for about 20 years when I came to his lab as a really rookie. From the third year of physical therapy, knowing nothing about science, really nothing about science. And Mike took me every step in this very committed way to become, whatever I'm here today. And I think that Mike, for me, is mentor with capital M, influencing, I think, more or less all decisions or all junctions in my life, apart maybe to who I should be married. But apart of it, Mike was really a central figure in my scientific career. But it's not about me today, it's about Mike. So Mike Gutnick he studied veterinary medicine in UC Davis. He then did his PhD at Stanford with David Prince, and he was graduated in 1972. He made in the same year, he made Aliyah, and joined Hebrew U in 1972, where he worked with Pat Wall, and he contributed a lot to our understanding of mechanisms of phantom pain. Then in 1974, in the Faculty of Medicine, Ben-Gurion University was open. Mike moved to Beersheba, to South, and joined the Faculty of Medicine. He was the founder of the Department of Physiology there. In 1997, he came back to Hebrew University to be a head of a veterinary school, and now he's in the Faculty of Agriculture. So during his scientific career, Mike published seminal papers in a variety of fields. Just mentioned some of them. It's very important papers exploring the intrinsic properties of cortical neurons and the biophysical mechanisms of epileptogenesis. He studied a lot of role of sodium channels modulation in epilepsy. He was one of the pioneers of studying sodium influxes, the way of sodium imaging in axonal initial segment, characterizing the mechanism of action potential generation. And when I joined Mike's lab, he became interested in the interlaminar circuitry of the gate neurons of the cortex, laminar layer 4 somatosensory neurons, and this is the project Mike is going to talk today. Please, Mike.
So Alex was wrong. I did tell him who to marry. And I was right. On the other hand, I'm not sure we ever agreed on who to vote for. But I once came to Alex and said, Alex, I read someplace that something like 75% of the Russians believe in transfer. What does it mean? He said it means that 25% didn't understand the question. Okay, today I'm going to talk about layer 4 of the barrel cortex. And the work that I'm going to tell you about is basically the PhD thesis of Omer Revach. And Alex and I are co-manchim of Omer. And you'll see why, because part of this work is in a sense a continuation of Alex's PhD thesis in my lab about 10 years ago. And the work is in collaboration with a group with a theoretical group in Germany, in Göttingen, with Andreas Neef and Fred Wolf. And basically, the fundamental basis of the work comes both from stuff that was done here in this department, and also stuff, theoretical work that was done in Germany. So to start off, I want to show you a movie. And this is a movie from the laboratory of Matt Diamond in Italy. And this rat, this is a rat, I think. But this rat, he reaches over here and he gets a piece of sandpaper. And the sandpaper can have one of two grates, very, very small. And what the animal does is he has to decide whether to turn right or left depending on distinguishing how rough the sandpaper is. And the animals are very good at this. They learn it very quickly. They can distinguish it very fast. You can see that he doesn't spend much time making his decision. In fact, from the time that his whisker touches the sandpaper to the time that he makes the motor decision and starts moving in a particular direction, on the average, something like 97 milliseconds passes. This information is processed extremely rapidly. And so it's got to get from the whiskers up into the cortex. It's got to get into the cortical circuit, and then whatever the cortical circuit's going to do the barrel circuit's going to do in order to tell the animal to do this. So, yes. Maybe it's not cortical. I think that he showed that it is cortical. Absolutely correct, because there's lots of places for interaction, subcortical interaction and whatnot. But the basis, I think what he showed was that the basis for deciding on size, for making that decision, is in fact dependent on the cortical circuit. There are lots of things about, because this is a complicated system, the cell is whisking and whatnot. So there are lots of places where the sensory and motor can interact. That's true. So here's the summary of what I'm going to talk to you about. Layer 4 of the barrel cortex, like the visual cortex, layer 4 has these spiny stellate cells. In the barrel cortex, it's most of these cells. They're very small. They're the only excitatory neurons in this area of the cortex which begin and end in the cortex. They don't send any, and in fact, they don't send any or almost no axons out of the barrel itself. And they constitute kind of a main gateway into this barrel circuit. There are other pathways into the circuit. The thalamocortical input goes to other layers as well, but what I'll show you is that these cells only really are interested in the thalamocortical input. Theoretical studies, which set me off on this thing, theoretical studies suggest that because these spiny stellate neurons are very small, the theoretical studies were from him, okay? Because these cells are very small, their dynamic range should be very limited. And because their dynamic range is limited, they should be slower. And the question is, if they're slower, if they're so tiny, and they are tiny, they should have a limited capacity to track high-frequency input. And we measured for the first time how fast these cells can track an input, and indeed, they're very slow. They're not fast, as expected. But yet, their response to whisker stimulation is quite rapid. And so what I want to talk to you today is about how fast processing is achieved, how they've managed to process very quickly. And what we show is that the unique properties of their synaptic background noise makes them fast. And it's what makes layer 4 able to relay sensory information into the cortical circuit as a whole. So first, I don't think that these days that anybody has to talk too much about introduce the barrel circuit, but you know, these are the whisker field is plotted onto the contralateral cortex, whisker by whisker, and there's a pathway, a direct pathway through the brainstem to the thalamus, and from the thalamus up into the barrel cortex. So that for any point in the cortex is related to any single whisker. And so the thalamus, VPM in this case, we'll talk only about this fast through pathway, the lemniscal pathway. So the VPM projects, and it projects into layer 4, projects into layer 6. It also projects quite a bit into layer 5, and the layer 5 neurons receive quite a bit of input also. However, a standard layer 4 neuron has an input resistance on the order of 300 or more megohms, And a standard layer 5 neuron has an input resistance well under 100 megohms, and so an equivalent current will cause very, very much less of a voltage deflection in a layer 5 cell. On top of that, what you will see is that these layer 4 neurons, they get 15%, 13 to 15%. People have counted these things religiously. 13 to 15% of their excitatory input comes from the thalamus. That's a huge amount, actually, because the cells only have about 900 to 1,000, 1,500 synapses onto them. And so this large amount of their input comes directly from the thalamus, and the rest of their excitatory input comes almost entirely from other layer 4 neurons in the same barrel. So they're talking to each other, and they're taking and relaying information from the thalamus. This information then gets relayed to all the layers, but primarily into layer 2, 3. And there is a huge convergence, so that every layer 2, 3 cell is getting a large proportion of its input from every layer, from every spiny stellate neuron in the barrel. About half of them, half the spiny stellate neurons in a given barrel are projecting onto a single, onto every single layer 2, 3 neuron. Okay. So the thing about these cells is that they're tiny, they're spiny, they are confined to the barrel, and their axon trajectories go up into layer 2, 3 and down into layer 5, but mainly up into layer 2, 3. They're small, excitatory, vastly interconnected, confined to the barrel. This is what's said. Their main target are thalamocortical axons. They distribute their output to all of the layers. So I think it's fair to say that layer 4 is a relay layer, that its purpose is somehow to bounce thalamic input into this circuit in some sort of a coherent fashion. So along came Guy and said that these tiny cells, this is a layer, this is scaled drawing of what a spiny stellate neuron looks like and what a layer 5 neuron looks like from the mouse cortex. You can see that these are tiny cells, and that should have a strong impact on the encoding capabilities of the axon. And the question is, how can these cells then fire so rapidly? So, in order to show you how we did it, this is done by experiment by Peterson in vivo. Watch this this guy's whiskers. When he begins to whisk, this is recording from 2 cells simultaneously in layer 4. When he begins to whisk, note that the synchronous activity decreases tremendously. And there's, in each cell, there's kind of an asynchronous activity, and he quantified this, so that, and the cross-correlation between any 2 cells goes down considerably when the animal is whisking. And so, this is a kind of gives an idea about how this the general mode of the way we think about it may be. We say that, okay, let's say that there is asynchronous input onto the axon, onto the dendrites, and that then, as a result, the cell fires spikes on this asynchronous input. So what we can do is we can put in a fluctuating input current. And, my colleagues in Gutnick developed the algorithms to generate an asynchronous input in which which is random, but has defined characteristics, defined temporal characteristics. And we can define the time characteristics of the input current. So our experiment in general is to catch a cell. We work in slices. To catch a cell, to inject a current, and to bring it close to threshold, to get it firing at about 5 hertz, okay? And then to relate, and then I'll show you how we then look at the dynamic characteristics of the spike train as it relates to the input. So the first thing that we wanted to ask, this just shows you the cross-correlation and how it goes way down when the animal is actually in a position to be looking for stimuli. So we take this fluctuating input current, we put in a fluctuating input current, and then on this fluctuating input current, we superimpose a very, very tiny sinusoid. And we ask for each frequency of sinusoid how good the neuron is when it's firing at about 5 hertz, which is what it fires at when it's feeling the periphery. When it's firing about 5 hertz, how well or precisely timed are those spikes at the with the various frequencies we're putting in. So, if we take and we put in this current, and we take time characteristics, let's say, of correlation characteristics of 5 milliseconds as the autocorrelation time, then what we see is that a layer 4 cell, what we're looking at here is the firing rate as a function of time, and time, of course, is the thing. When we look at 50 hertz, the two cells, a layer 5 cell or a layer 4 spiny stellate cell, are equivalent. But at 200 hertz, the layer 5 cell follows it very, very nicely, but the layer 4 cell has got a lot of problems following at 200 hertz. So this bears out the concept that this tiny cell, the smaller cell, in fact is a bit of a low-pass filter. And if we look at the vector strength, which is just asking, just the number giving us the precision with which the spike is spiked versus the peak of the sine wave, what we can see is that the layer 4 cell falls off at frequencies above 100, whereas the layer 5 cell can follow frequencies that are much faster than that. So spiny stellate neurons are worse than pyramidal neurons at encoding high frequencies. But then there's another set of studies which claim that the synaptic noise and the time characteristics of the synaptic noise should have an effect on the filtering, or on the, and that if the synaptic noise is slower, that in fact it will broaden the dynamic range of the cell. And these are basically theoretical studies. Brunel was the first one to publish this stuff, and Fred Wolf has been a champion of this. And so we decided that we would try and see what the actual synaptic noise looks like in these cells. Now, looking at the actual synaptic noise, we have to look at it in current. We have to look at the current noise, because otherwise we'll just be trapped by the time constant of the membrane. And to look at the current noise is very difficult. What we found was that we had to use electrodes with input access resistances under 10 meg all the time. These are tiny little cells, and it was fortunately they're graduate students and they have to get their degree, and so Omer poured a lot of blood down the electrodes to get these data. It can't be done in vivo. First place, we could never get access resistances that low in vivo. The other reason it can't be really done in vivo is because in vivo there's too much spontaneous activity going on anyway, much of it synchronized, that would keep us from doing an experiment. So what Omer decided to do was he took a light-activated caged glutamate. He bathed the barrel cortex in this caged glutamate, and then he shined UV light way off the center, so that it caused a barrage of activity inside of the whole thing. We waited so that the effect of the glutamate that was actually released by the light is minimal, and then we were able to collect and see over time what the noise characteristics are. Of the caged glutamate, you can see here. For instance, this is the shining of the light, and then you get this wave of activity, which then disappears in like about 200 milliseconds, because we're constantly perfusing the slice, and so we're washing it out.
So you wait for that?
No, we wait from here to here, right? We wait to the peak. But we're fairly far away from where the light was shined. And when we did this experiment in layer 4 and in layer 5, what we found was that layer 4 has slower noise. So this is the autocorrelograms of the noise in layer 5 is in blue, and in orange is of layer 4. And what you can see is that the correlation time wasn't that much slower, but it was somewhat slower. It was a difference of between about 2 to 3 milliseconds versus 8 to 10 milliseconds. No, in current noise. In current noise itself. So we just measured at the soma. Everything is measured at the soma. Right. So it could be that the noise in the pyramidal cells is a little bit faster because of space-time errors. That could be. Yeah. Okay. When you take and you record from one of these cells in slices, they're silent. There's almost no synaptic noise. They're silent, they don't speak, they don't talk. Okay? What we did was we measured the noise that was generated by these cells when we tried to grind up the circuit. Since most of the cells are connected to each other, I expect that most of what we saw here is the activity of nearby layer 4 cells. Well, but we're in a slice. So a lot of other sources just aren't seeing anything. Right, which is mostly coming from the other layer 4 cells. It's the excitatory input to the cell. It's also inhibitory input to the cell, by the way. No, it's got nothing. I cannot define which synapses are giving this noise. I'm just saying, what's the background of this thing? I'm also not pretending that this is what it looks like in vivo, okay, where there are many other sources of input to these cells probably and whatnot. But this is the closest we can come to asking.
Chris, you can't induce it electrically or oxygen, just in a cleaner way where you don't have the potential for this additional glutamate to be running around in the system?
We don't mind the additional glutamate because basically we shine the light here, we record here, we shine the light for a very brief period of time, and the additional glutamate is all caged. To the glutamate itself. No, we don't. And if we look at the PSCs that are generated by this noise, what we can see is that the layer 4 PSCs are slower than the layer 5 PSCs. By the way, so the layer 4 PSCs are they decay much slower than the layer 5 PSCs. And we knew that this was going to happen because that was Alex Beachstock's PhD thesis 10 years ago, or 10 years before this, actually. I don't know how long ago, but a long time. Because we had seen before a puzzling thing. We had recorded from layer 4 cells, and what we had seen, working with Alex and with Ilya Sledovich, who's now in Beersheba, what we had seen is that the layer 4 cells, the spiny stellate cells, have a distinct kind of NMDA channel. And it's distinct in that it's activated at resting potential. So NMDA channels are slower, as you know. They introduce calcium. That's another thing that they're famous for. And usually their function in plasticity has to do with the fact that they need depolarization to relieve a magnesium block. But these, which are NR2C subunit, they don't need that depolarization. So that a lot, not all, but a lot of the NMDA current that is present, or a lot of the spontaneous synaptic current that's present in layer 4 cells is in fact much slower and is mediated by NMDA channels. And here you can just see from those days, that in layer 5, for instance, if you look at minus 70 millivolts, you see some activity which is entirely removed by an AMPA blocker, and when you depolarize, then you see the NMDA activity. And it's not entirely removed by an AMPA blocker at resting potential in spiny stellate cells. So this was always puzzling to us because we couldn't figure out why this current would be there, why this NMDA channel would be there. There's good evidence that there's almost no plasticity in the input in layer 4. This, after a critical period, plasticity, the synaptic plasticity seems to disappear. And we couldn't see where calcium would have much of an effect. And so we were very pleased to see that these cells are slower, but not by very much. And when I talked to our theoretical partners, they said that they would hypothesize that this slowness wouldn't be enough to make very much of a difference based on their calculations. So we decided to see whether it would make a difference. And from now on, I'll show you recordings from spiny stellate neurons. So this is looking, what we take is spiny stellate neuron, and we inject it either with very fast, with very slow noise, 10 millisecond correlation time, or very fast noise, 2 millisecond correlation time. In each case, we move the electrode, or we move the membrane potential up so the cell is firing about 5 hertz, and we ask the question, how good is the cell, the spiny stellate cell, how good is it at following? And what we can see is that at 200 hertz, the fast correlation time is much worse than the slow correlation time. So I think that that's pretty obvious. And if we look here at the vector strength, which is a measure of the precision, what we can see is that this is the same cell with a fast or with slow. And you can see that there is a big difference in their dynamic range, even though this is a very, very small difference in the time correlation of the membrane. Now, another way that we can see this is instead of putting in sine waves, we can just looked at a Fourier transform of the noise and compare it with a cross correlate the Fourier transform of the noise with the spikes and get a gain curve, essentially a gain curve, and because it's a spike train, basically the thing is to just look at the spike triggered average and look at the characteristics of the noise and spike triggered average. And what we can see is that this is the two kinds of recording from the same kind of cell. Okay, from the same cell, and we can see that we get the same result, these two different ways of checking the dynamic range of the cell. So these cells now are faster because the noise is slower. So now we can ask the question, how fast then can a layer 4 can a circuit of layer 4 cells, a population of layer 4 cells, respond? Remember that each layer 2-3 cell is hearing from many layer 4 cells. So if one doesn't fire, another one will fire, okay? Or another one. And so what we can do is we can divide up our train and say that with each sweep, basically we're looking at a different cell. Because the cells are very, very similar one to the other. And so we can now, by the number of sweeps, we can make a sort of an estimate on the number of neurons that would be necessary to cause a significant change. For the population of spikes to actually recognize that something happened. So what we did was we embedded in this population of spikes a tiny EPSC of 20 picoamperes. And this number we got because of studies which have shown that with single stimulation of single thalamocortical fibers, that's the mean size of the EPSC that is recorded in a spiny stellate neuron. So this is like the minimum response that the cell can have, that this system can have. And so we put a synchronized input of this little PSC into, we repeat it again and again and again. We do sweep after sweep. And we then measure when the population of spikes is statistically different, which is clear that the population has seen that something happened. For some reason, I changed all this, and it came back the way it was before. I guess I'd like it to be that way, but no, it's okay. The way that I changed it was that this is where we put in the synchronous activity. These are spikes, populations of spikes that we record from the soma. For a variety of reasons, we're pretty sure that the spike we record in the soma, we can detect him at the soma 1 millisecond after the initial segment where the spike was actually generated and which we can't sample, made the decision to spike. So basically, there's a delay here of 1 millisecond that comes from that delay back propagation of the spike into the cell. So this one is corrected, and what you can see is that with 900 sweeps or 900 neurons, there is a clear identification that something has happened, whereas with the fast noise there is not, and this is with many, many more neurons, 2,700 neurons or 2,700 sweeps. Still, the fast noise in this cell can really determine that something has happened. And So this is a relevant population size of layer 4 cells based on a variety of measures. And what we can see is that the detection probability goes up tremendously when the noise is slower. And we showed you before that the noise actually is slower. So now leaves us one question. So what we know now is that these cells have slower noise, and we know that the slower noise confers on them the ability to be much, much faster, as fast as pyramidal cells in detecting this thalamic input. The question is why? Why would slower noise do this? And again, I asked the computational people why, because they're the ones who came up with this. And they're also the ones who said that this small difference shouldn't make much difference at all. And they couldn't tell me, well, they could tell me why, I just couldn't understand it. But what we decided is that there must be some molecular mechanism for this. There's some reason why slow noise would work. And one mechanism we could think of is that slow noise can open more potassium channels. And when more potassium channels are open, a couple of things happen. And one of the things is that there's a decrease in resistance, input resistance. And so you should get two effects. One is a higher threshold. But the other thing that you should see is a much more faster rise of the spike at its point of origin. And again, there are several studies, including from Brunel and from others, which have shown, and this is just a few of them, many studies have shown, that in fact, the neuronal response to fluctuating inputs is determined by the rate of rise of the spike at the site of its origin. Now, in these cells, in all cortical cells, as far as I know, including inhibitory neurons, the spikes originate not in the soma, but in the axon. So this was first discovered or described by Brock, Clement, and Eccles many, many years ago in 1953, where that there is this initial segment and that this is the site of spike generation. And in cortical neurons, we see it very, very clearly. This is in the derivative of the spike, you can see this hump on the initial thing, which is the spike as it occurs in the initial segment before it backpropagates in soma. And in phase plots, it's very, very obvious. This early part here is definitely being generated out in the initial segment. And just to show you what that looks like, this is a cell in which we've injected a sodium dye. So it's a dye which is very sensitive to sodium concentration. Sodium doesn't bind to anything except this dye, but it's pretty freely diffusible. The Kd of this dye is much above the sodium concentration in the soma. And if we look here, we can see, is it working? No. Okay. You can see over here the spike, and once there's a spike, you can see that the spike is really generated and there's a huge increase in sodium concentration in the initial segment. The initial segment is the site of spike generation. And the initial segment of cortical neurons is really a very, very interesting, structure. It's almost as complicated as a synapse in terms of its protein structure. And it's got a very complicated protein skeleton. And the important thing is that it binds, that the cytoskeleton binds various ion channels and concentrates various ion channels at this point. So there's sodium channel binding, calcium channels are bound there by ankyrin G, and several kinds of potassium channels are bound there, and particularly the M current channel and a KV1 channel, which is another potassium. So the KCNQ channel is the M current, and this is another potassium current, voltage-dependent potassium current. Both of these currents are activated by depolarization. And here you can see that this is looking at the M current, and the other is the ankyrin G. Ankyrin G is the protein marker for the initial segment, and you can see that both the KV1, KV1.2, KV1.1, and the M current, all of these channels are present and concentrated in the initial segment of the cell. And what this shows is that when we use a blocker of KV1 channel or a blocker of M current, this effect of slow current totally disappears. That is to say, indeed, the effect is because the slow current is activating potassium channel. What we think is happening can be seen here. This is just looking at spikes in the same cell when generated either by 10 milliseconds or by 2 milliseconds. And what you can see is the shift in voltage. And then if we look now at the phase plots, we can see that, in fact, the threshold is shifted in a depolarizing direction by about 2 millivolts or so. But there's a sort of a slow depolarization here, and the slow depolarization is what is necessary to activate potassium channels, which is shifting the voltage, but it's doing something else. The other thing that it's doing, this is a sodium activation curve from one of these cells. And if we look on the sodium activation curve, that shift is enough to shift us considerably on the rising phase of the sodium activation, And because we're shifted on the rising phase, it means that the spike should rise much faster in the initial segment. Now, we don't have a probe right now to go into the initial segment and see and measure what the difference in spike timing is, the rate of rise is, but everything fits with the idea that it is much faster. You will recall that cells, for instance, in the auditory system, which need to rise very fast because they follow at tremendous speeds, inputs at tremendous speeds, the cells have very, very low input resistances. You put in a current pulse, you almost get a square wave out rather than get a slow thing. They've got very, very slow. And so this is why we think that the slower noise is compensating for the fact that these are such tiny cells. So in summary, the anatomy shows us that spiny stellate cells listen to the outside world and relay the information into the cortical circuit. Because the neurons express an unusual NMDA channel, the background noise fluctuates slightly slower, that the time correlation of the background noise is slower. This small change leads to a dramatic increase in the ability of these cells to respond rapidly and reliably to input. The capacity to encode high frequencies explains how a limited number of layer 4 neurons, which are prone to low-pass filtering, can still do their job, can still pass within a millisecond, or relay the information coming from the thalamus into the rest of the circuit. And finally, this increased capacity to encode high frequencies is lost when the potassium currents are blocked. So, thanks a lot. We were doing the simulation. We haven't made figures of it yet, but we've done a couple of simulations by putting in the potassium and raising. It does fit the time course of activation of the potassium channels, and these potassium channels are really segregated to the initial segment. This we have yet to model, but I expect that it will. Because there are lots of models which show that increasing the rate of rise of the spike, in fact, has a huge effect on the dynamic range of the cell. Okay, well, thanks very much.